Hi everyone, my name is Lanchantin and I'll be presenting our work Transfer Learning for predicting virus-host protein interactions for novel virus sequences. This is joint work with Tom Weingarten, Arshdeep Sekhon, Clint and Yanjun. Proteins are essential biomolecules that carry out critical functions within our bodies. So they do things like oxygen transportation, there's antibodies, and digestive enzymes. Proteins are a chain of molecules called amino acids that are linked by covalent bonds. There are 20 different amino acids, allowing us to represent proteins as a string with an alphabet of 20 different characters. So each protein can be viewed as a string that's typically around 500 characters long. The key aspect about proteins is that the string of amino acids folds first into a local secondary structure and then into its full 3D tertiary structure. And this structure is what determines the function or role of a protein. However, proteins rarely act alone, and instead they interact with other proteins in order to carry out specific functions. So in order to determine what a protein really does, we must first know who it interacts with. And this is critically important, especially when studying viruses, because viruses infect humans through protein-protein interactions. So for example, the COVID-19 spike protein interacts with the human ACE2 protein in order to enter human cells and replicate. And while we know certain interactions with human proteins for previously discovered virus proteins, we want to know which human proteins a new virus protein will interact with. The problem here is that obtaining protein structure in interaction partners is slow, expensive, and hard to replicate. On the other hand, sequencing technology has allowed us to sequence proteins quickly and cheaply. So our goal is to accelerate protein interaction discovery and understanding by predicting interaction partners directly from sequence in silico. Well, many works have shown that it's possible to accurately predict protein-protein interactions from sequence. So given two protein sequences, the goal is to predict the likelihood that they interact with each other. But few works have focused on the task of predicting interactions of a novel virus. And this is due to the fact that when it comes to novel virus-human interaction prediction, there's two main problems. The first is that limited interaction data is available for a novel virus. And this is because we assume that the proteins are just discovered for a novel virus and we don't have interaction information available for those proteins or possibly for many similar proteins. The second issue is that interactions are determined in part by structure, so predicting only from sequence may not be sufficient. So to handle the limited data problem, it's been shown in other domains that leveraging large amounts of unlabeled data in the form of self-supervised training can help on downstream tasks with smaller amounts of labeled data. To handle the problem of predicting interactions from sequence where structure information is important, it's been shown that protein structure can be predicted directly from sequence data. So in this work, we introduced Deep VHPPI, which is a transfer learning framework for sequence-based protein interaction prediction. So this framework includes two pre-training steps before fine-tuning the model on interaction prediction. So the first step is a mass language model, which is a self-supervised training procedure trained on millions of unlabeled Swiss protein sequences, where the objective is to predict matched amino acids given their context. And this helps the model learn general protein representations that can help on downstream tasks. The second step is to further pre-train the model on a supervised structure prediction task. So this is a multi-task setup to predict secondary structure, contact, and remote homology. So this helps the model learn protein structure information that can be used for the downstream interaction task. Well, finally, we fine-tune the model to predict the interactions between two proteins. So for this, we feed each protein through the model before a classifier predicts the probability that the two proteins interact with each other. So we have a virus protein and a human protein, and the classifier gives an interaction prediction probability. So this is trained using binary cross-entropy loss function. Putting this all together, we have one shared model that is used in a three-step training procedure for protein interaction prediction from sequence. While our framework is general for any sequence-based model architecture, we use a variant of the transformer. The transformers were first introduced in the context of natural language to model the relational structure between input tokens. So they're particularly useful in examples where the meaning of one token depends on the meaning of distant tokens. So in this example, the word bat refers to the animal due to the word flying. However, if flying is changed to swung, then the word bat likely refers to a baseball bat. The transformers were designed to model the context of words dependent on others. We're motivated to use transformers by the fact that protein sequences contain long-range context-specific dependencies that change the meaning of each amino acid. And transformers have proven effective for protein sequences by many recent works. The main problem is that when using character or amino acid tokens, transformers may have difficulty learning sequence motifs, which are short patterns analogous to words in English, which influence protein interactions. So to this end, we introduce a variant of the transformer model, which uses various convolutional filter widths in the first layer to learn these sort of word representations. So each different filter learns a different size amino acid word. And the transformer layers are these vanilla transformer self-attention layers to model the interactions between the words. So a sequence-based computational model to predict protein interactions can be utilized for two main use cases. The first being predicting novel interactions, and the second being analyzing mutation effects. So this first use case, where we assume a novel virus has just been discovered in sequence, we want to predict all possible human interactions. So we trained our model on about 250,000 interaction pairs from the HPIDB dataset, where 22,000 of these pairs are true or positive interactions and the rest are negative. And we tested the human interactions of proteins from 3 different viruses, including SARS-CoV-2, which are not used during training. So across the 3 different types of viruses, we saw performance gains using our approach compared to previous methods. And importantly, we showed that our transfer learning approach helps in all 3 testing datasets. So the second use case of a computational model to predict protein interactions is where we do know some interactions, and we want to predict how mutations affect these interactions. This is a particularly pressing problem in understanding how virus variants, such as the recent spike protein variants for the coronavirus, can change interactions. So we might have the original interaction score for the reference virus sequence, but we want to see what happens if there's a change in the sequence, or a mutation. So we might have the original interaction score, and we can run this simulation on any mutated sequence to observe the predicted effects. So to do this computationally, we observe the prediction value for a reference virus sequence, and then we manually mutate the sequence and feed it to the model again, and observe the change in interaction prediction. So we used 105 mutated spike sequences with their corresponding ACE2 binding affinities, and we trained the model on a subset of the mutated spike sequences and ACE2 and test on the remaining. So we simulate 3 different settings of labeled sequence pairs to use as training. So we're using 100, 1,000, or 10,000 different sequence pairs as training, and we test on the remaining. We evaluate this setting using testing sequences by comparing the binding predictions with the corresponding dissociation constant binding affinity. So as the number of training mutation sequences increased, we saw that the Spearman rank correlation with the ground truth dissociation constant also increased. So our model is able to learn the relationships between certain mutations and their actual binding affinities with ACE2. So to summarize, in this work we introduce a transfer learning framework for predicting protein-protein interactions between a novel virus and humans. We validate this model in 2 use cases, predicting novel virus interactions, as well as analyzing how mutations affect interactions. And with that, I want to thank my co-authors for helping make this work possible.